So, by the way, I'm Gnaneshwar. I am a postdoctoral fellow at National Center for Biological Sciences in Bangalore. Today, I'll be talking to you about RNA biology as an emerging target in drug discovery. Let's understand this very interesting area and an up and emerging area in drug discovery. And let's understand why it is relevant in today's session. So I'm very sure most of you will have read these books as part of your curriculum in the colleges that you're studying and the courses that you're taking. Some of these are Principles of Genetics by Robert H. Tamarin and Principles of Genetics by Snostad and Sammons, and many other similar books, right? So these are what give us a basic understanding of the biology, the genetics, and the flow of information in biology. So while we understand the basics from our textbooks and our courses, it's very important to keep up with the developments in the field, which are rapidly progressing. And as the pandemic came and left us with some scars and also with some knowledge about how to deal with biology of things around us, we can focus in today's session to understand some of these concepts, right? So what these books essentially teach us is fundamentals such as Mendelian genetics, where we know Gregor Mendel was very instrumental in discovering the origin of these traits in plants that he was studying, right? So he circled back on certain elements in our body, in our cells, that could have the basis for the traits that living organisms carry, right? So the basis of such traits was then later traced to DNA, whose structure was unraveled by these gentlemen, Francus and Crick. However, there was another person who immensely contributed to this same study done by Francus and Crick, which in fact won them Nobel Prize. But one significant contribution from this person was left out from such a recognition. Just a question to you guys. Can you tell me who this person is in this image, and what was their contribution, as you might see here to your right? You can send me your response in the chat box. This is just a simple quiz to know. Yeah, Watson and Crick are these two gentlemen here, but who is this person? Who is this lady who also contributed immensely to this same study that won these people the Nobel Prize? Yes, one of you got it right. It's Rosalind Franklin. It was her data of X-ray crystallography of the DNA that led these gentlemen to unravel its structure. So critics, and this is my personal opinion too, she too deserved this recognition, this Nobel Award, along with these gentlemen, because she provided key data which these gentlemen used to unravel the structure of DNA, right? So such controversies always exist, and people move on in the field, but such mistakes are not to be repeated. That is the point. So next, we had one of our own who from the Indian origin, who was working in the United States at the time, Hargobind Khurana, who immensely contributed to the structure and sequence of the DNA, right? So it started from Mendel saying that we have certain elements that are responsible for traits that all of us carry, and these elements everybody figured out is DNA, and DNA structure was known. Now came what is in the DNA, right? DNA is nothing but these 4-letter codes, T or U, T in DNA, U in RNA, right? And DNA carries T, TCAG or UCAG in RNA. These are 4-letter codes that are dispersed throughout our nucleic acid sequences, be it DNA or RNA. And the way these come together in triplets give rise to codes for amino acids like U, and U creates phenylalanine. U, C, and U creates serine, right? And so on. The rest of the table tells you the whole story of what code gives rise to what amino acid. This is what we must have all studied in our fundamental learnings of genetics, right? And for this, Hargobind Khurana was also awarded the Nobel Prize because it is a key discovery which tells us what sequence in what order can give rise to what amino acid, and this is the code that our DNA and RNA carry, right? So DNA is a double helical structure. RNA is a mostly single single linear fragment, right? So this is our understanding. So let me tell you that the world has discovered so much beyond these triplets of codes and what proteins and amino acids, etc., are being coded by our body. We have literally understood what is the whole genetic makeup of human beings and many other species, and what are these codes that give rise to different protein sequences, and what are these proteins as such, right? So the field has moved on a lot. In simple words, you can categorize the field into few basic levels, where you refer to this as genome, wherever the genes are present. Sorry for this. Wherever the genes are interspersed on the sequence of DNA in this double helical structure, you have genes that are interspersed here, and these genes present on DNA is referred to as the genome. And these genes get transcribed into RNA from the DNA. RNA is formed, which are referred to as transcripts, right? Transcription is the process in which the DNA is transcribed into RNA. So RNA is referred to as the transcriptome, where there is largely ribosomal RNA, which is not going to code for any protein. Here you have messenger RNA, which codes for proteins on the ribosome, right? So you have messages from your genes on DNA that have transcribed as mRNA, which gets translated into proteins on complexes known as ribosomes. And these ribosomes are made of ribosomal RNA, also referred to as rRNA, which make these ribosomes along with some proteins. We'll come to that in a while. So rRNA is forms the major component of the transcriptome, much more than mRNA, right? So rRNA forms 95% of what gets transcribed as RNA from DNA, 95%, and only 5% or less than 5% is mRNA. The rest of it is non-coding RNA, which doesn't code for any protein sequence. And this RNA world is very interesting. This is referred to as the RNA world. This RNA world is very interesting. What we have learned very well from our textbooks is that these messenger RNAs are very well translated into proteins on these ribosomes, where mRNA forms the proteins, right? And this layer of information is called proteome, where there are different proteins that are being translated on these ribosomes from different mRNA that form a part of transcriptome. And this transcriptome is transcribed by genome. So this is the way the genetic information flows from DNA into RNA into protein, right? Slowly, the information from genes trickle down in the cells to form proteins which build the cell, which serves as the building block for cells, that builds the cell, maintains the cell, and regulates the cell and dictates the function of the cell, right? So this is essentially the way genetic information flows from genome to transcriptome to translatome to proteome. By the way, this is a new terminology that might not be there in many textbooks, which is referred to as translatome, right? What is translatome is essentially the mRNA that is translating into protein. You need to keep in mind that not all messenger RNA, just because they are made in the cells from the DNA, not all messenger RNA get to translate into proteins. Only some portion of mRNA translate the message into proteins. The rest are just stuck in different pockets of the cell, waiting for signals to take them to translation, right? So not all mRNA will be translating, and only those mRNA that are translating on ribosome is referred to as translatome. Okay, these are different omic layers which are important for regulating development and disease. Okay, so in simple words, this translatome refers to only this mRNA that is translating into proteins. This is not the translatome. This is the transcriptome here. Okay, and not all of it will translate into proteins. This fundamental understanding just developed from the last decade or two. So earlier people thought genes make RNA, and these RNA all get translated into proteins. But later on, people figured out that there is a bottleneck in this flow of genetic information. This bottleneck was very visible because people did sequencing, next generation sequencing of this transcriptome, and they did proteomic profiling as well through mass spectrometry, where they can read and count proteins. And in next generation sequencing, you can read and count the RNA. Both data were not matching with each other. So they understood that there might be a bottleneck somewhere in between, from transcriptome becoming proteome. There must be a bottleneck. And when they looked at the messages that were actually translating, that told them what portion of this transcriptome is actually translating into protein. So all of this, the reason I'm telling you is because this whole control, this whole regulation happens in this small tiny world, which we think is tiny, but it is a vast ocean of information and data, where you will find it in this RNA world. And people have been trying to get to the bottom of these details to understand disease, development, and how to make better drugs to target and treat diseases, right? So the reason I say that this is the bottleneck, right, and this bottleneck is part of the RNA world, is because of this. You see, these two subunits are ribosomes. Okay, ribosomes are made in the nucleolus and they come out into the cytoplasm where the mRNA, this strand of mRNA, gets attached to these subunits of ribosome, from where the translation happens, and these translate mRNA into proteins, right? They keep on translating. If you look at what these ribosomes are made of, which I have already told you, ribosomes are made of 80 different ribosomal proteins and 4 different ribosomal RNA. And these ribosomal RNA, also referred to as rRNA, that become a part of ribosome, are one of the major components of the RNA world, where the RNA world, 95% of it is made of ribosomal RNA. And these are huge pieces of RNA running up to 2 to 3 kilobases, right? 2000 to 3000 kilobases, right? These are huge pieces of RNA and present in large numbers in the cells, more than your mRNA and other kinds of RNA. So the trick here is that these RNA regulate more RNA such as mRNA and help them translate into proteins, which we have learned are very important to make what we know as our cells. And these proteins are important for the form and function of the cell, right? For such an important function, the RNA, the ribosomal RNA, is very important, as you can see. And what happens here is this ribosomal RNA combines with different ribosomal proteins, 80 different ribosomal proteins. And this whole assembly is done by 200 odd factors for this to become one complex and start translating mRNA into proteins. Now, you will appreciate this. Such a complicated organelle, such a complicated, complex organelle is going to make protein from your mRNA. 200 factors are just required to build this. And this whole complex is made of 80 different proteins and RNA coming together to make these wobbly balls of subunits, which become one ribosome that actually translate the message into proteins. So this can be regulated by changes in any of these factors. If you want to control translation normally or abnormally, you have 80 different proteins, 4 different ribosomal RNA, 200 factors. Not all the time they will behave the same way, in the same manner, for this to be enabled. That is the point I want to drive. And people have seen that many of these factors go haywire in many diseases, which are not genetic diseases per se. They go haywire due to epigenetic deregulations. Now, let's not worry too much about that. What I mean to say here is, it's a complex process, as you can see here. I just used a simple cartoon to illustrate that ribosomes translate mRNA into proteins. But if you break it down into its skeleton of information, you will see all of these intimidating numbers of components that come in to participate in making proteins, right? We assume this information flow to be linear, but this is the bottleneck where ribosomes can restrict what can be transcribed, what can be translated into proteins, right? It can happen because of deregulation in any of these factors, like these initiation factors, these assembly factors, these elongation factors, termination factors, anything. So all of these information, take it that it is gibberish for now. It is the idea is to just intimidate you and make you aware that this is not a simple process where complicated arrangement of molecules and assembly and processes in play are very essential for messenger RNA to be translated into proteins, where you might have learned about tRNA, where each tRNA will carry one amino acid, and there will be several such tRNA which carries particular amino acid, right? So in this process, you can imagine this as a factory where proteins are being made in the cells, where this factory requires nearly 1,000 components, right? So if among these 1,000 components, the RNA, the ribosomal RNA itself is the major component, what can happen to protein synthesis, even if there is minor upset in ribosomal RNA, right? You can imagine what will be happening to the production process, protein production process in this factory, right? So that is the power of the RNA world, which the world has realized very late because of late advances in this area. So now, having said this, let's go back to this chart where you have the schemes of information that is flowing, right? Genome to transcriptome to translatome to proteome, right? And here I said the RNA world plays a huge role in many different ways. Let's understand what is this RNA world even made of, right? So the RNA world is made of certain RNA that is coding in nature, meaning that they carry the genetic code in mRNA to make proteins, right? So that is why they are called the coding RNA. Whatever is non-coding, this is simple to understand, right? Are you with me? Tell me if you understand this part, the coding RNA part. Just say yes or no. Okay, great. So the coding RNA part is clear. It is simple. It carries the information from DNA on RNA, which has the code in the messenger RNA. That is why you call it as messenger RNA, which translates into proteins, right? Those are the coding RNA.
It's 17 hours.
Now you have the non-coding RNA, which has several different categories. One of which is ribosomal RNA itself, which I told you is the largest of the population in the RNA world by size and volume. Okay, and there is non-coding RNA, which is pretty ambiguous because rRNA is also non-coding and tRNA is also non-coding. And in the non-coding RNA category, you have small RNA and long coding RNAs. Okay, and here you have piRNA and miRNAs. And in the long non-coding RNA section, you have intronic long non-coding RNA, long non-coding RNA, enhancer long non-coding RNA, circular long non-coding RNA, and long intergenic non-coding RNA. So there are different categories of RNA which were discovered with advances in sequencing technology. As the sequencing technology kept pace with advances, initially people started sequencing DNA and then they started sequencing RNA. They added more power to the RNA sequencing through next generation sequencing, where lengths and kinds of RNA that could be sequenced were challenged through decades of efforts. And with those advances, you captured a lot of data which tells you what this RNA world is actually made of, beyond just messenger RNA that we all study in our textbooks, right? So there is a whole area of the RNA world that is still waiting to be tapped for information and characterizing their function. So I can say, even with the advances that we have, we know very little of RNA, their use, their function, and how we can use it for our therapeutic value, right? So this is one. And now the non-coding RNA, let's understand more details about these non-coding RNA. There is long non-coding RNA and small non-coding RNA and circular RNAs here. Long non-coding RNAs, okay, LNC RNA, are greater than 200 nucleotide in length. So they are called long because they are greater than 200 nucleotide, and this is the limit they have set to call something as long non-coding. Anything shorter than this is called small non-coding RNA. And here in the small non-coding RNA, there are also rRNAs, which are also small, some of them. tRNAs are also small, and small nucleolar RNA, which are present in the nucleolus, right? So they are important for certain functional roles where they carry an information locally to either DNA or to other RNA or to protein, right? And play a functional role in carrying the information or in modifying something at the site, at the target site, okay? So they are functional in role. And there is a regulatory role, such as microRNA, which regulate other RNA or protein availability. siRNA, which works very similar to miRNA. Both of them regulate the availability of messenger or other kinds of RNA. And then there is piRNA, quasi-RNA, tells RNA. All of these are regulatory in their roles. And there are the long non-coding RNA are very important in terms of these classifications, such as they are classified based on structure, action, and location, right? When it comes to structure, they are either linear or circular, okay? So this is a very important distinction. Don't worry about memorizing these other terminologies, which actually we will break it down into fundamental details in the detailed session that we are planning, to which I'll come to in the end. So getting back to the basics quickly, these long non-coding RNA are either linear, meaning straight linear non-coding RNA, or circular non-coding RNA. And they have a role to play, such as cisRNA, cRNA, transRNA, right? And they are classified based on locations as well. Some of these might be intergenic, bidirectional, where the RNA could have meaning in both the directions, right? And intronic RNA, those RNA that are present in the introns, sense and antisense strand containing RNA, right? So basically there are categories of RNA that are characterized based on function, regulation, structure, action, or location. That's what is important to understand from these. And what happens here? What is the role of all these non-coding RNA? In simple is this, right? So this is the RNA world that we are interested in dissecting and understanding. And this RNA world influences the genes, right? They play a role in epigenetic regulation of the genes that are present on DNA. These RNA, some of the non-coding RNA, can go and bind to certain DNA sequences based on sense, antisense sequences, based on sequence complementarity. They can go and bind to certain target sequences in DNA and help genes in those sequences turn on or off by attracting certain proteins that help in regulating those DNA sequences to either turn on or off, right? So that is one of the roles of non-coding RNA, which regulate the genome. And they in turn regulate themselves. This non-coding RNA can regulate other non-coding RNA in their own world, where they have some sequence complementarity. Like some of you might have heard about microRNA, right? The microRNA will have some sequence complementary to its mRNA targets, right? So when they have complementary sequences that match mRNA target, they go bind there and they attract certain proteins that destroy this mRNA or block its translation, right? They regulate the mRNA translation, some of the non-coding RNA, meaning that this world regulates its own kind in this population of RNA, right? So they regulate themselves. So non-coding RNA can regulate genome, they regulate the transcripts, they even regulate translation, the event of translation here, where ribosomes are to translate messages into proteins. These non-coding RNA can bind to ribosome or messenger RNA and interfere or enhance their ability to translate and make proteins, right? They are regulating this world as well, the translatomic layer of information as well, right? And finally, they can interact with proteins and regulate the function of protein, which is to either build the cell or interact with the genome, transcriptome, or translatome, right? So this world can literally interact with outside with molecules outside its own kind, or it can interact with its own kind to regulate form and function of these molecules, right? This is very interesting. So has this been taught so far in any of your courses? Can you please say yes or no? This is just to gauge from you all where the field stands in terms of course content in curriculums that is present in the colleges. So this is just for my understanding. Let me know if this role of non-coding RNA has been taught. Okay, no worries. So that's fine. No worries. So this must add value to your understanding. So do stay with me till the end of this session and do register for this course so that you gain more insight as to where the field stands and how important this is for employment. We will come to it in the end of this session. Okay, so now this non-coding world can regulate literally anything in the cell, meaning that it has numerous downstream applications as well, right? So now let's switch our gears and go back to basics, right? What Mendel and whatever textbooks tell us is that genes are these elements that make us, and when I say they make us, I mean the traits. It could be good or bad, which dictates our health or disease states. From health, you can appreciate and learn. From disease, you can find treatment by understanding what health means in terms of biology, right? This knowledge is very important for applying it to treatment, right? If you know what health is, and if you know what disease is, and if you know the molecules that you are to play with to switch the sides as if you're playing on a seesaw, then you have understood the game and you are applying the game for your benefit. Now, on top of this, environment, develop, age, vulnerability adds certain changes to these pathways that are present in this, where your health can swing into disease state or disease can be swung back into healthy state, right? So basically, the textbooks are right that genes are the basis of our biology, but they are solely not the basis of our biology, right? They are still the basis of our biology. Our textbooks are still right. What I'm adding is upgradation of knowledge here through this course, where you will understand more details than what the textbooks have taught about genes and the flow of information, right? They can be influenced, these genes can be influenced by environmental and other external factors, which is fascinating, right? Like this Himalayan rabbit. The genes of this Himalayan rabbit make it have pigments when it is grown at when it is kept at 20 degrees or less. Switch the temperature to 21, you will already see it forming certain pigments, right? And rear it about 30, all the pigments are gone, right? So this is how important the biology is, not just understanding this as the sole basis. There are a lot of things that regulate us and elude our understanding still. Now, in line with basics, you must have learned that mutations, right, where one of your nucleotide sequence getting converted into other. It could be transition from pyrimidine to pyrimidine or purine to purine, or transversion, where pyrimidine becomes a purine or purine becomes a pyrimidine, like this, right? These base substitutions keep on happening, and they dictate whether we are healthy or unhealthy, whether our offspring is healthy or unhealthy, right? Like in the sickle cell disease, one amino acid change from glutamine to valine at a significant position in the protein can determine whether somebody will be carrying sickle cell disease or not, right? So this is how important it is. And just to revise basics, there is normal sequence. This is the normal healthy sequence where AUG codes for methionine, GCC codes for alanine, and so on, right? This is the normal code. Now there is a silent mutation which doesn't impact the code much, because GCT and GCC still code for alanine. Now there is a nonsense mutation where TGC becomes TGA. TGA is stop codon. So the protein abruptly stops. This is a serious condition, right? And then there are missense, where one amino acid changes can happen. Frame shift, where the frames tend to change because of just one nucleotide deletion or insertion deletion, etc., etc. Don't go by this information. This is just for you to read while I am telling you these basics. I've already told you these basics. It's just the same information as what I told you. So this is the genetic basis of disease, where mutations give rise to diseases, right? This is our simple understanding. Now, the best way to deal with these kind of mutations is sorry, the best way to deal with these kind of mutations is to edit. Now we have Crispr technology, which can edit the genes and correct the mutation. With Crispr technology, you can undo any of these mutations, right? Undo whatever the mutation that has happened that is causing disease. You can reverse the disease by gene therapy using Crispr or other gene editing tools, right? So to do that, your target is the DNA. And targeting DNA in gene therapy is a very complicated process, right? Because see, what is happening in gene therapy is you are editing the DNA, basically, which is at the top of this chain of information. If you edit the DNA, the information flow is regulated here, is tuned in a way you want it back, right? But DNA editing has its own issues and lack of control at certain places. So editing the central code for life is very complicated and in many cases irreversible. What if you edit using gene therapy and somewhere down the lane, you understand that it is very problematic? Can you reverse the gene therapy, right? Such reversals are very difficult, or you will have to take care of this human being who has been gene edited permanently at his embryonic stage. By the way, embryonic gene editing is still prohibited in the country and in the world too, right? But the problem with gene therapy is that once you edit in the embryo, it becomes a human, and the edited information passes on to the progeny through sperms and eggs, right? It becomes a permanent edit. And to reverse that, you need to do reverse gene editing if there is a problem, right? In somatic gene therapy, what is happening is you take the cells of the patient outside the body, edit the genes in those cells and put it back inside the body. But it is temporary because cells are going to only proliferate so far, right? And it is not permanent, but it solely depends on the population of the cells that are propagating, which have been gene edited. So you are at the mercy of their propagation here, right? So this is ex vivo. By ex vivo, I mean, again, somatic gene therapy, where things are edited outside and put back. Or in vivo is the same thing, right? So in either case, you are meddling with the genes of a human being or any animal where you have very little control. You don't have enough handle, right? If something goes wrong, because you have thousands of genes in the body, and your gene therapy has every chance of not just affecting one gene, but multiple genes, right? So it is a very risky area of research to target genes, to target the DNA in gene therapy. But nonetheless, it is one of the areas which has made significant progress. But there are shortcomings in this area where it cannot cater to all diseases and all patients, right? Now, coming back to the RNA world. The RNA world, as we learned, also plays a role in a lot of diseases, not just in development and maintaining cells. If they go wrong, there are a lot of diseases that are also caused by RNA defects. Some of these are cancers. Most of them are, frequently occurring cancers such as pancreatic, AML, acute myeloid leukemia, right? AML, and chronic myeloid leukemia, lung, liver cancer, different cancers, name it, there is an RNA component that is defective in these diseases as well. Similar with one of the most challenging areas of drug discovery, which is neurodegeneration, where it has a lot of failure. None of these diseases I list here still have a single therapeutic molecule, right? So neurodegeneration affects memory and learning. It's happening in Alzheimer's, Parkinson's, Huntington's, autism, where the brain degenerates or cells in the central nervous system degenerate, right? And in autism, there isn't much degeneration, but it's a it's a learning, memory and learning associated disease. Okay, so all of these have RNA and RNA as a component in causing disease. Okay, now there is HIV/AIDS, where there is also a RNA component, and there are several rare genetic disorders too, where there are RNA, that are targets and biomarkers. Okay, so having said that, where do we stand in application of all this knowledge, right? So right now, the years of pandemic from 2020 brought us close to understanding the application of some of these elements from the RNA world, where we now talk about RNA and using them as vaccines, right? So you have closely experienced the pandemic. All of us have closely experienced the pandemic and the progress the science made during the pandemic. And one of the progresses was coming up with an mRNA-based vaccine for COVID-19, right? And this mRNA-based vaccine is lauded as one of the most significant breakthroughs in science, where you started using RNA as vaccine rather than protein based vaccines that have been there from early 1950s or 60s, right? So we now talk about RNA based vaccines, right? So in this mRNA vaccine for COVID-19, let's just understand what happened in terms of its design and progress. So what they did is, I'll not go into the basics of COVID-19, right? We all understand what a spike protein is. Even my family members be it my mother, father, or brother, or even friends, do talk about these spike proteins. Spike proteins are basically those spike-like elements present on the virus, which the virus uses to bind to our cells, right? The virus, okay, let's just see if I have that slide. Let's see if I have that slide. Yeah. So here, these are the spike proteins that I'm talking about, right? So the spike protein binds to our cell receptors, ACE2 receptor. So the spike protein is a key component where the interaction between the virus and our cells happen, right? So you should either take care of ACE2, but if you take care of ACE2, our cells are going to suffer. If you take care of some element that the spike protein or something that is important for its interaction with our cells, then this process of virus invading our cells will not happen. That is our basic understanding, right? So now that spike protein is what is used as vaccine in many of the vaccines. But the clever hack that mRNA vaccine guys did is use the mRNA of the spike protein. This mRNA that makes the spike protein was encapsulated in a lipid nanoparticle for it to be carried into our cells, so that this mRNA now makes protein, which is nothing but the spike protein, right? These mRNA make this viral spike protein in our cells, and our cells raise immunity against the viral protein that these mRNA vaccines make, compared to the protein that is injected as vaccine in other kinds of vaccines, right? So this is one advantage of mRNA based vaccine, where you inject mRNA that makes the protein that becomes the vaccine. So you call this the mRNA based vaccine. And here the RNA world, the understanding from the RNA world had told these mRNA vaccine researchers that this uracil can be converted into pseudoiridine, and if you do that, if the uracil is converted into pseudoiridine in RNA, this will not cause toxicity to cells, right? Our cells are usually finding uracil rich RNA as toxic. If you inject RNA into our cells, they find uracil containing RNA as toxic. So over decades of research had told that uracil is the component that is causing toxicity. So they modified the uracil into pseudoiridine. Wherever the uracil was there, it is converted into pseudoiridine in these mRNA vaccines, and this doesn't elicit adverse immune reactions which are toxic to our body, right? So this this mRNA based vaccine didn't come to be just because they decided one fine day, okay, why not inject mRNA and make protein that become the vaccine, right? So it dates back to late 1987, when Robert Malone, who performed certain landmark experiments in the field, where if you happen to think of it now, it looks very simple, right? But at that era where even sequencing was in its early stages, what he did was he copied these messenger RNA. These messenger RNA were copied and they were made in the lab, and he mixed these messenger RNA, mRNA, with droplets of lipids to encapsulate these messenger RNA. By the way, RNA is very unstable. RNA is one of the molecules in the cell which is very unstable. Lipid is temperature and light sensitive, but RNA is sensitive to everything: light, temperature, And RNA, as soon as you open a bottle of RNA, the RNA starts degrading at room temperature because of enzymes, RNases that are present everywhere. Microbes in the air have it. Our cells have RNases. RNases are present everywhere. These are nucleases, which okay. Somebody raised a hand. I can't see who. It's okay. So what you can do is, if you have questions, you can post it in the chat box. That will be easy for me to pick up and answer. Okay. Don't wait till the end. If you have any queries, do post it in the chat box. We can pick it up from there. Okay. Now getting back to. Okay. Sorry, there seems to be many people raising hands. May I know what is it in the chat box? Not able to find. Okay, if you don't have a question, please make sure you are not raising your hand. If you have any question, post it in the chat box or in the Q&A section. We can take it up towards the end. Okay, now let's go back to the slide. Yeah, so here I was saying that all of this mRNA vaccine research started not just recently during the pandemic. Most of the work for it was built from early 1980s. It started with the idea from Robert Malone, who mixed these messenger RNA with droplets of fat. And here I was referring to the unstable nature of RNA, where it can degrade quickly. It is not stable like a protein or DNA. DNA lasts for millions of years, right? You can even extract DNA from artifacts that you excavate from all these archaeological sites. But RNA is not that stable. RNA degrades very quickly. The reason is the enzymes, the nucleases that degrade RNA are present everywhere in the microbes around us and in our own cells. So you need to protect the RNA from such contaminants reaching the RNA, right? So one of the ways is to encapsulate it in droplets of fat, which Robert Malone tried. This not just protects the RNA from enzymes, but also helps in delivery as well, which is very important for any RNA vaccine to be delivered inside the cell, for the cells to absorb these RNA inside the cell bodies. You need to encapsulate these RNA with lipid droplets, right? So having done that, he bathed this combo and checked if human cells can absorb such mRNA encapsulated with fat. And he saw that these cells do it, and they began producing proteins from these messenger RNA inside those cells, which is a key breakthrough from where many other discoveries followed. But it is important to notice that he is the first who wrote, after seeing his data and his results, he wrote in his diary that RNA could be targeted for drugs in the future, right? Which is a very important finding that actually translated into efforts in 2020, 21, and 22 through mRNA vaccines, where millions of people around the world got these mRNA-based COVID-19 vaccines, and the sales of these are expected to reach US $50 billion in 2021, which we have already crossed. So yeah, This in itself speaks volume of these early simple experiments that started way back in 1980s, right? So now let's understand the history of mRNA vaccines. There is a saying that a journey of 1,000 miles begins with that first step. So Robert Malone took that first step where he actually encapsulated these mRNA in lipids and delivered it to cells, right? But even before that, mRNA had to be discovered, and then proof that mRNA makes the proteins, tested in the lab, and then Robert Malone happened, where he synthesized mRNA in the lab, covered it with proteins, lipids, and delivered into cells to make proteins. Next, this strategy was tested in rats. So whatever you test on cells, next you test it in animals, right? In rats, mice, or primates. And this is where long time in drug development takes, where you keep testing the drugs on animals for a long time to get as much information as possible for its effects. Excuse me. Now here, there were several findings, right? Where the actual experiments didn't just start on rats, they tried it on frog embryos, right? They first tried it on human cells, and then tried it on frog embryos, and then tried it in rats and mice. And there were several vaccine strategies that were tested, such as mRNA vaccines tested for influenza itself in mice. And these happened through 1990s, right? 1990s to 2000. And then first report of 4-component lipid nanoparticles that are actually efficient in delivering and efficiently eliciting an immune response, right? So they discovered that modified RNA with pseudoiridylation can evade immune detection for toxicity, but can be used for raising the desired immune reaction, which is based on and which is very specific to vaccine, right? So the toxicity could be non-specific. The immune reaction that is specific to vaccine is what you are going after, which is very specific to those diseases or viruses or whatever infection, right? So you want to achieve specific effect but overcome non-specific effect, and this non-specific effect was overcome by modifying the RNA using pseudoiridylation and other hacks, right? And also delivering these mRNA using lipid nanoparticle. In 2000s is when the nanotechnology emerged as one of the solutions for many of the drug discovery problems, one of which was delivery of your drugs to the target site, right? So that was taken care. And then they tested several vaccines such as rabies vaccine, etc., etc. And then in 2020, there was a chance to try this whole strategy to translate it and apply in patients through mRNA-based vaccine. And this is the vaccine that actually found prominence and application in the society, which saved millions of people by giving them immunity against COVID-19, right? So this particular saga is about the mRNA vaccine alone. But just to develop a quick understanding of where the field stands, let's look at this beautiful demonstration by this company Laronde, which makes these RNA that are circular, okay, which they refer to as eRNA, right? So these eRNAs are circular, and let's understand from their video how these eRNAs are very important. So let's quickly understand what these eRNAs are capable of doing, right? So these eRNAs enable in vivo, meaning inside the animal, expression of any protein. It is programmable, persistent, modular, tunable, and continuously redosable, right? Because it is circular in nature, this eRNA, what they refer to as endless RNA, right? So this circular loop of RNA is a simple hack which these people developed for endless translation of protein, right? Let's understand from their beautiful video what this actually means.
Traditionally, proteins are encoded and made from messenger RNA. In order to be expressed as a protein, mRNA engages with a protein factory called the ribosome. This interaction is initiated by a cap on one of the ends of the mRNA. There are other naturally occurring forms of RNA called circular RNAs. These do not have ends and are unable to translate into proteins. Other forms of RNAs have evolved to initiate translation without a cap, like RNA viruses, which do so by utilizing a ribosomal engagement site called IRIS. What if we could incorporate such an element into circular RNAs to take advantage of their unique characteristics and make them translate into proteins? By synthetically designing a circular RNA with an IRIS, we were able to initiate continuous translation, and eRNA was born, an endless translatable RNA.
Right? So in essence, what they have achieved here is a circular RNA by incorporating the viral IRIS site on this RNA to make the ribosome translate and keep looping around and endlessly translate this protein, right? This protein, in our case, what if it is a COVID-19 vaccine protein, right? If this protein is the COVID-19 vaccine protein, which can be endlessly translated, you probably don't have to take booster doses, right? Because in small amounts, your body is producing this protein at times. This is hypothetical. There is no such vaccine. Let me caution you here. This is just hypothetical. This is just an idea I'm proposing here as a solution to overcome this issue of booster doses, right? Because we have all heard that these vaccines that we have received might lose their efficiency, our system might lose the memory of these vaccines, and so on and so forth, right? So to periodically keep boosting ourselves with this vaccine, what if we had an endless mRNA COVID-19 vaccine, which could give us booster doses on its own in our cells like this, in this format, right? This is just a hot and emerging idea which this company has developed, not for the sake of vaccine delivery, but for making certain proteins that are lost in diseases, right? Certain proteins that are lost in diseases need to be made continuously by the cells. So to churn out those proteins continuously that are lost in diseases, they developed this eRNA platform, which will make this endless translating RNA to replenish those lost proteins. So this is just one example of the advances that is made with respect to drug discovery targeting RNA, right? Besides the COVID-19 vaccine that we learned about. Similarly, there are companies, right? There are companies there are companies, some of them are very popular, some of them big, some of them small, some of them scaling up at all levels. So currently, there are companies that are interested in developing drugs targeting RNA. Some of these are Takeda, Eli Lilly, which are big names in the pharma industry. Microsoft and Calico are surprising entries into this area of drug research, and more specifically, they are interested in RNA targeting drugs, which is very important to notice, right? So there is a lot of potential for RNA based drug discovery that is going to very soon hit the market, and the demand for employment might also increase in these areas. And Calico is Google's arm for life science research. So Calico is also focused on RNA-based drugs. Microsoft is focused on RNA-based drugs. The advantage that companies like Google and Microsoft, which are into computers basically, is that they have enormous capacity to look at data. The data that is already there in the field about RNA that is listed in public databases. So these companies might have sophisticated algorithms which can detect or simulate certain drugs that can be used to target these RNA, right? So RNA is just one target for these companies, and if they set their mind on the right target with right resources, they can design efficient drugs. And that is something that must be happening. What is happening beyond their walls is only known to them, because most of these are trade secrets. Okay, now there is another company known as Anima Biotech. We can have a look at Anima Biotech also as a case study for how the field has emerged and how the field is developing. So this will be one last stop for us to understand what is happening in the field by diving right into it. Okay, we will do that. But meanwhile, there are other companies that you can see here that have formed in very recent years. Some of them have very unique selling points, such as there is a company that is known for focusing on drug development, keeping tRNA as their platform, right? We looked at Laronde now, and there are other companies here like Ribometrics, Arrakis, Revire, etc., etc. These are promising companies that are expanding in drug development focused on RNA. So these are all global players, and India is not going to be left behind in this race. Indian companies are also going to take this advantage of RNA information that is out there in the world and leverage it for drug discovery. So it is soon happening, and there is a potential opportunity here that is bound to amplify. So let's now take a look at this company called Anima Biotech and understand what they are doing. Sorry. So while this website opens, let me check in the chat box if there are any queries. Okay, none. So we speak about mRNA translation, right, into proteins. So what this company is doing is they are using high-scale phenotypic screening on their platform called mRNA Lightning platform, right? So don't be lost in in the visuals that is there in the circus of things. What is happening here is they have a technology where it will light up the cells when the translation of a particular protein happens. As simple as that. When a protein translates, it lights up, right? That's the signal that these cells give. These computers, these sophisticated set of computers on these high content imaging, which images those signals and then records, and these robotic devices automate this whole process of treating these cells with drugs. So basically they have cells that they are treating with drugs, and they have a platform on which the cells light up when the protein translates, right? Or not translates, then the light turns off, vice versa. So they treat it with drugs and test whether these drugs have an effect on these disease target proteins. And they keep on testing drugs, thousands of drugs, in this high content imaging analysis that they keep doing, and their computer constantly keeps monitoring what drug affected what protein synthesis. And by doing that, they have a lot of drugs in their pipeline that they have figured out, right? So this is the technology that I'm talking about, where the mRNA translation can be recorded as light signals which the drugs might influence or not influence. And this, they look at the effect on mRNA, right? And they are focusing on mRNA biology that is getting influenced by these drugs and how it influences the translation, right? And how they can leverage this content in discovering drugs for these diseases such as lung fibrosis and other fibrotic conditions, CMYC related tumors, MCRAS related tumors. They're even working on Alzheimer's, pain, repeat associated diseases, etc., etc. And they are doing great work building significantly molecules in their pipeline, where they have their own molecules and molecules that they have partnered with big companies to discover. So all of these are new and emerging areas with very interesting players that are coming up with novel technologies to discover drugs in this, right? So the course content that we are planning will have A to Z of these information. One is start with basics always, which is understanding fundamentals in the flow of genetic information. Then we dive into the RNA world, and then we look at the ribonucleoprotein complexes, then look at the mRNA and translation. We understand different non-coding RNA, microRNA, small interference RNA, small non-coding RNA, SNO-RNA, small nucleolar RNA, pvRNA, and then other different non-coding RNA that we studied here, and how they regulate genome, transcriptome, translatome, proteome, etc., right? And then we understand RNA and diseases associated with RNA in detail. We will dive into targets, therapeutics, treatment. We look at biomarkers and disease targets, etc., right? And we look at drugs such as chemical molecules. It could be antisense oligos or just chemical molecules, which are large and small molecules alike, that are working on these targets. And we look at biomolecules such as microRNA, RNA itself, that could be used as vaccine or a treatment, therapeutic, gene therapy, RNA-based gene therapy, or CRISPR, right? And we can look at applications and patents that are there in the field to understand. And we could add more content meanwhile. This is the initial content that we have discussed to cover in this course, right? So what happens here with this with this course is that there are several places where you will need to use this knowledge and experience. It could be academia, such as in universities and research institutes, or in industry, right? Industry being the pharma biotech industry, where your experience or inexperience will determine whether you get that first offer to train or to even join them, right? And there could be an opportunity provided or denied because you are inexperienced or your experience is not sufficient. So you could either be fortunate by taking up these courses, or unfortunately miss the experience that you gain from these courses. Some of it is definitely fixable. Unfixable is when you may not be able to engage in such an enriching content. So I urge you all to fix this knowledge gap that currently exists in our curriculums. Go extracurricular and take this course and fix this knowledge gap in a mentored and eventful period, right? By taking this course, which will help you get closer to where you will apply in academia and industry. With this course that you take, you can gain knowledge, some sort of dry lab experience, confidence, because you have had knowledge and some experience, you obviously feel confident and closer to your, sorry, this is not gene editing, this is your RNA drug discovery job, right? So this is a possibility. So thank you all. I urge you all